morphine, naloxone, and buprenorphine can all interact with the μ-opioid receptor, yet their effects are remarkably different. Morphine activates the receptor as a full agonist, naloxone binds without activating it and competitively blocks opioid agonist signaling. Buprenorphine activates the receptor but with lower efficacy as a partial agonist. So how can ligands interacting with the same receptor produce such different outcomes? Welcome to PharmaVault. In this video, we'll build a clear scientific framework for understanding how drugs interact with receptors and why binding alone cannot tell us what a drug will do. The answer depends not only on whether a drug binds, but on what happens after binding. Not every drug acts on a receptor. Before focusing on receptors, we need to correct one common oversimplification. Not every drug acts through a receptor. A drug target is the molecular or sometimes physicochemical entity through which a drug produces its effect. Salbutamol is a receptor-acting drug. It activates beta-2 adrenoceptors in airway smooth muscle, promoting bronchodilation. Aspirin acts primarily through enzyme inhibition. It irreversibly acetylates cyclooxygenase enzymes, thereby reducing the formation of prostanoids. In platelets, inhibition of cyclooxygenase 1 reduces thromboxane A2 production and platelet aggregation. Lidocaine blocks voltage-gated sodium channels, reducing the initiation and propagation of action potentials. Fluoxetine inhibits the serotonin transporter, reducing serotonin reuptake. Antacids demonstrate an even clearer non-receptor mechanism. They chemically neutralize gastric acid without requiring a conventional biological receptor. So, receptors are extremely important drug targets, but they are only one part of pharmacology. From this point forward, we'll focus specifically on receptor-mediated drug action. What is a receptor? A receptor is a biological macromolecule, usually a protein, that selectively recognizes ligands and through which those ligands can influence biological signaling or function. Receptor selectivity is relative rather than absolute. A receptor preferentially recognizes ligands with compatible chemical and structural properties. But this does not necessarily mean that only one molecule can bind. Cell surface receptors interact with extracellular ligands and transmit information across the plasma membrane. This is especially important for ligands that cannot readily cross the lipid bilayer. Intracellular receptors are located in the cytoplasm or nucleus and interact with ligands capable of reaching those compartments, often but not exclusively lipid-soluble molecules. The receptor's location, molecular structure, and method of signal transmission allow us to organize receptors into four major pharmacological families. Four major receptor families. The first family is the ligand-gated ion channels, also called ionotropic receptors. In this family, the ligand-binding receptor and the ion-conducting pore are parts of the same molecular complex. Ligand binding directly changes channel opening and therefore ion movement across the membrane. Because the receptor does not require a long intracellular signaling cascade, responses can begin within milliseconds. This makes these receptors particularly important in rapid synaptic transmission. Examples include nicotinic acetylcholine receptors, GABA-A receptors, and 5-HT3 receptors, serotonin 3 receptors. Different members conduct different ions. Nicotinic and serotonin 3 receptors are primarily cation channels. while GABA-A receptors are primarily anion channels. The second family is the G-protein coupled receptors, or GPCRs. These receptors contain 7 membranes spanning alpha helices. Their activation can regulate heterotrimeric G proteins, second messenger systems, ion channels, enzymes, and other signaling proteins. Because signal transduction involves intermediate molecules, the characteristic onset is generally seconds to minutes, although some GPCR-mediated events can begin more rapidly and downstream effects may persist much longer. Examples include beta adrenoreceptors, muscarinic acetylcholine receptors, dopamine receptors, and opioid receptors. The third family includes enzyme-linked and kinase-linked receptors. These are usually single-pass membrane receptors. Some possess intrinsic catalytic activity. The insulin receptor, for example, has intrinsic receptor tyrosine kinase activity. Other receptors do not possess their own enzyme activity but associate with intracellular kinases. Cytokine receptors commonly signal through associated Janus kinase. The JAK-STAT pathway is an important example. Initial phosphorylation events may occur within seconds to minutes, while downstream effects on metabolism, growth, differentiation, or gene expression can develop over minutes to hours. The fourth family consists of intracellular and nuclear receptors. Their ligands must be capable of reaching the cytoplasm or nucleus. Classical activation produces a ligand-receptor complex that regulates gene transcription. Glucocorticoid receptors are commonly located in the cytoplasm before activation and then move into the nucleus. Thyroid hormone receptors, in contrast, are generally located in the nucleus and associated with DNA. Because gene transcription and protein synthesis take time, classical genomic effects usually develop over hours to days. However, more rapid non-genomic effects have also been described. These time ranges are useful comparisons, not absolute rules. The measured speed depends on the receptor subtype, cell system, signaling pathway, and the particular endpoint being observed. Orthosteric versus allosteric binding. After identifying the receptor family, our next question is, where on the receptor does the ligand bind? The orthosteric site is the receptor site recognized by the endogenous physiological ligand. An orthosteric drug binds to the same site or an overlapping site and may compete with the endogenous ligand. But orthosteric does not mean agonist. An orthosteric ligand may activate the receptor, partially activate it, reduce constitutive activity, or block the actions of another ligand. Orthosteric therefore describes binding location, not functional effect. An allosteric ligand binds to a topographically distinct site. By altering receptor conformation, it can modify the affinity and/or efficacy of an orthosteric ligand. A positive allosteric modulator, or PAM, enhances the action of the orthosteric ligand in the system being studied. A negative allosteric modulator, or NAM, reduces it. The diagrams show baseline, enhanced, and reduced responses in the same direction. The NAM is reducing the magnitude of the response, not necessarily generating an opposite biological effect. The GABA-A receptor provides an important clinical example. GABA binds to orthosteric site on the receptor. Diazepam binds to a separate benzodiazepine allosteric site on benzodiazepine-sensitive GABA-A receptors. Diazepam acts as a positive allosteric modulator. It enhances the receptor's response to GABA, but it does not simply replace GABA at its orthosteric site. Classical benzodiazepine modulation requires GABA to be present. This gives us an important rule. Binding location and functional effect are separate questions. Now that we know where a ligand can bind, we can ask two further questions. How readily does it bind, and what does it do once it is bound? This brings us to affinity and efficacy. We have established where a ligand can bind. Now we need to separate two properties that are frequently confused: affinity and efficacy. Affinity answers the binding question. How readily does a ligand bind to a particular receptor? Affinity reflects the molecular interaction between the ligand and its binding site. A higher affinity ligand generally favors receptor occupancy at a lower free ligand concentration than a lower affinity ligand under comparable conditions. But affinity tells us only about binding. It does not tell us what the ligand will do after binding. That is the role of efficacy. Efficacy answers the activation question. Once the ligand is bound, how effectively can the ligand-receptor complex generate signaling? A ligand with agonist efficacy stabilizes receptor states associated with signaling. A partial agonist generates a lower maximal response than a higher efficacy agonist in the same receptor system. A neutral antagonist can have substantial affinity while possessing no intrinsic activating efficacy. This distinction brings us back to our opening example. Morphine binds to the μ-opioid receptor and produces agonist signaling. Naloxone also binds, often with high affinity, but does not intrinsically activate the receptor. Buprenorphine has high affinity and activates the receptor, but with lower efficacy than a full agonist under the same conditions. Efficacy must also be interpreted within a biological system. The observed response depends on a receptor density, coupling efficacy, signal amplification, cell type, and the endpoint being measured. Finally, pharmacological efficacy is not the same as clinical effectiveness. Here, efficacy refers specifically to the ability of a ligand-receptor complex to generate a pharmacological response. So remember, affinity tells us whether binding is favored. Efficacy tells us what the bound ligand does. A ligand can bind very tightly and still produce no receptor activation. The efficacy spectrum. This conceptual concentration response graph shows 4 positions on the efficacy spectrum. The dashed horizontal line represents basal or constitutive receptor activity. Some receptors can spontaneously adopt active conformations and produce a degree of signaling even when no agonist is bound. A full agonist has positive efficacy and can produce the maximal response available in that particular experimental system. As its concentration increases, the response rises until it reaches a plateau. A partial agonist also has positive efficacy, but its maximum is lower. Importantly, this lower maximum remains even when all accessible receptors are occupied. Adding more partial agonists cannot convert it into a full agonist. A neutral antagonist behaves differently. When administered alone, it remains at the basal activity line because it has affinity but no efficacy. It does not increase or decrease constitutive activity. Its effect becomes apparent when it blocks the action of an agonist, or in a constitutively active system, an inverse agonist. An inverse agonist has negative efficacy. It preferentially stabilizes inactive receptor state and reduces constitutive signaling below the basal level. Inverse agonism can therefore only be observed when the receptor system has measurable constitutive activity. Without basal signaling, a neutral antagonist and an inverse agonist may initially appear similar. The curves here are conceptual. We are comparing the direction and maximum of the responses, not using their horizontal positions to compare affinity or potency. Those quantitative relationships deserve a separate video. Another important point is that the observed maximum is not determined by the ligand alone. Receptor density, receptor reserve, coupling efficiency, and downstream signal amplification can all influence the response measured in a particular tissue. The central message is that efficacy describes the effect produced by the ligand-receptor complex, positive, absent, or negative. How antagonists block receptors? First, a terminology clarification. The statement affinity without efficacy applies specifically to a neutral antagonist. An inverse agonist, as we just saw, has negative efficacy. Neutral receptor antagonists can produce blockade through different mechanisms. On the left, we have reversible competitive antagonism. Here, the agonist and antagonist compete for the same orthosteric binding site. Because antagonist binding is reversible, increasing the agonist concentration increases the probability that the agonist will occupy the receptor. The antagonist can therefore be outcompeted. This is called surmountable antagonism. Naloxone at μ-opioid receptors and atropine at muscarinic receptors are familiar examples of drugs that commonly behave as reversible competitive antagonists. On the right, we have insurmountable blockade. This means that simply adding more agonists cannot fully restore the original response. However, two different mechanisms can produce this pattern, and they should not be treated as identical. An irreversible orthosteric antagonist binds at the agonist site but dissociates extremely slowly or forms a covalent bond. The affected receptors are therefore unavailable to the agonist. Phenoxybenzamine is a classic example because it binds irreversibly to an alpha adrenoceptors. A non-competitive antagonist prevents normal receptor activation through a different mechanism. It may bind to a distinct regulatory site, or in the case of some ligand-gated ion channels, block the channel after activation. Because it does not simply compete reversibly for the agonist site, increasing agonist concentration cannot reliably overcome the inhibition. Both mechanisms may reduce the maximum response available to the agonist, but there is an important complication: spare receptors. If a tissue contains more receptors than are required to produce its maximal response, some receptors can be inactivated without an immediate reduction in the observed maximum. Only after the receptor reserve is exhausted does the loss become obvious. For this introductory video, remember the practical distinction. Reversible competitive blockade is generally surmountable. Irreversible or non-competitive blockade is generally insurmountable. This competition becomes especially interesting when the competing ligand is not a neutral antagonist, but a partial agonist. Why a partial agonist can oppose a full agonist? A partial agonist is still an agonist. When it is given alone, it binds to receptors, activates them, and produces a genuine biological response. Its defining feature is that the response is lower than that produced by a full agonist in the same system, even when the partial agonist occupies all accessible receptors. But what happens when a partial agonist and the full agonist are present together? If they bind to the same receptor site, they compete for receptor occupancy. Every receptor occupied by the partial agonist is a receptor that cannot simultaneously be occupied by the full agonist. Because the partial agonist produces less activation from each occupied receptor, replacing full agonist occupancy with partial agonist occupancy can reduce the total response compared with the response expected from the full agonist alone. That comparison is essential. The reduced response on the right side of the slide is relative to full agonist alone. It is not necessarily lower than the response produced by the partial agonist alone. The exact result depends on their concentrations, affinities, efficacies, and receptor occupancy. This is sometimes described as functional antagonism of the full agonist. But the partial agonist has not become a neutral antagonist. It continues to activate the receptor. Buprenorphine provides an important clinical example. It has high affinity and partial agonist activity at μ-opioid receptors. When administered alone, it produces opioid effects. Because it binds strongly and occupies a large proportion of these receptors, it can also limit the additional response produced by full agonists such as morphine or heroin. However, this should not be described as complete or absolute blockade. The clinical result depends on buprenorphine dose, receptor occupancy, prior opioid exposure, the full agonist concentration, and the specific effect being measured. This gives us the answer to the question from our opening slide. Two drugs may bind to the same receptor, yet produce very different outcomes because binding is only the first step. Their efficacy and their competition with other ligands determines the final response. Potency versus efficacy. Before we finish, there is one final distinction we need to make. Potency is not the same as efficacy. Potency asks how much of a drug is required to produce a defined effect. In this illustration, both drugs produce the same 50% response, but drug A achieves it at a lower concentration. Drug A is therefore more potent than drug B. A common experimental measure of potency is EC₅₀, the concentration that produces 50% of that drug's maximum response. A lower EC₅₀ generally indicates greater potency within the same experimental system. Efficacy asks a different question. What is the maximum response that the drug can produce? Drug C reaches a higher response ceiling than drug D, so drug C has greater efficacy in this system. This distinction is clinically important. A highly potent drug is not automatically more efficient, safer, or therapeutically superior. Potency may influence the concentration or dose required, while efficacy describes the response ceiling. For now, remember this simple comparison. Potency is about how much is required. Efficacy is about how much response is ultimately possible. Why receptor behavior matters clinically? Receptor pharmacology is clinically important because patients experience the consequences of binding, efficacy, and kinetics, not binding alone. First, receptor behavior influences therapeutic benefit. A ligand's efficacy contributes to the maximum response that can be achieved, although the observed effect also depends on the tissue, receptor density, and individual patient. Second, it influences safety. The same receptor system can mediate both therapeutic and harmful effects. Activation of the μ-opioid receptor, for example, can produce analgesia, but excessive activation can also cause potentially fatal respiratory depression. Third, affinity, occupancy, and competition influence drug interaction and reversal. Morphine activates μ-opioid receptors. Buprenorphine has high affinity but lower efficacy and can limit the response to additional full agonist. Naloxone competitively occupies these receptors and can rapidly reverse opioid effect. Finally, binding and activation are dynamic processes. Association, receptor activation, and dissociation kinetics can influence how quickly an effect begins and how long it persists. Recent research shows why this matters. In a 2024 study, researchers developed a negative allosteric modulator of the μ-opioid receptor. In animal experiments, it enhanced the action of low-dose naloxone against morphine and fentanyl-induced effects, while reducing withdrawal-related behavior. This is preclinical evidence, not an approved treatment, but it demonstrates an important principle. Understanding receptor conformation, affinity, and ligand kinetics can potentially improve the way medicines work in real clinical situations. Clinical pharmacology therefore depends on how strongly, how effectively, where, and for how long a receptor is influenced. Let us finish by collecting the essential ideas from this lesson. Key takeaways. Here are the 4 ideas I want you to remember. First, binding does not necessarily mean activation. After binding, a ligand may fully activate, partially activate, block, or reduce receptor signaling. Second, affinity, efficacy, and potency are different. They describe binding, the response produced after binding, and the concentration required for a defined effect. Third, ligands can have different functional roles. Full agonist, partial agonist, neutral antagonist, and inverse agonist do not produce the same receptor behavior. And finally, these differences have clinical consequences. They influence therapeutic benefit, adverse effect, interactions, reversal, onset, and duration. Understanding receptors is therefore one of the main connections between molecular pharmacology and the medicines patients actually receive. Thank you very much for watching, and I see you in the next lesson. In the next video, we will learn how to read concentration response curves.